this video, I'll be going over the immune system. Within the immune system, we have many different types of white blood cells which help protect us from pathogens. Another word for white blood cells is lymphocytes. They can be split into three categories: phagocytes, T cells, and B cells. Phagocytes' role is to engulf pathogens and destroy them. They do this through a process called phagocytosis, which I'll go through in detail later in this video. There are two types of phagocytes that you need to be aware of: macrophages and neutrophils. Then we have the T cells, which can either be T helper cells, which can help stimulate B cells production, hence they are helpful; cytotoxic T cells, which kill infected or damaged cells; and then memory T cells, which provide long-term protection against pathogens by remembering their antigens. Lastly, we have B cells, which can then be split into plasma cells, which can make antibodies, and B memory cells, similar to the T memory cells, which also provide long-term protection. Now, I understand that there are a lot of terms to remember here, so please make sure to wrap your head around these terms before your exams, as it will help you use these keywords in your application questions. So, our immune system can detect a pathogen using antigens. Antigens are proteins on the surface of cells which act as markers. All of our cells have antigens as well. However, our immune system detects our own antigens as self, but when a pathogen enters the immune system, it is able to detect their antigens as foreign. So, our body has many defense mechanisms against pathogens. The first line of defense are the physical barriers, the main one being our skin. Others include hydrochloric acid in our stomach, which provides the low pH that kills bacteria; gut and skin flora, which are good bacteria that normally are found in these places that compete with the pathogens for food and space. If pathogens breach the first line of defense, the body's second line of defense are the non-specific responses. These are not specific to any particular pathogen. Inflammation is where specific cells called mast cells are activated to release histamine, which cause vasodilation, dilating blood vessels, and increases the blood vessel permeability. Increasing permeability allows the white blood cells and antibodies to reach tissues. Lysozymes are enzymes found in tears and mucus, which kills bacterial cells by damaging their cell wall. Interferons produce protein within the infected cells to stop viral replication and increase the activity of cytotoxic T cells. Then we have blood clotting. As you have probably noticed, whenever you cut yourself, you will notice the body stops the cut from bleeding out. The reason for this is to prevent pathogens entering the body through that opening in the skin, hence makes a temporary seal before a scab forms. As I was saying before, phagocytes are involved in phagocytosis. There are 4 stages that you need to be aware of in your exam. This can be an easy long mark answer question. The first step is the phagocyte is attracted to the pathogen via chemotaxis, which is where chemicals released by the pathogen leads a path to it. Phagocyte engulfs the pathogen via endocytosis. Then the pathogen is trapped within the phagocyte's vacuole, which then fuses with the lysosome. When the vacuole and lysosome fuse within the phagocyte, it forms a phagosome. The lysozymes that are present will then destroy the pathogen. I hope you found this video useful, and please give this video a like. If you want to see more videos like this, then please subscribe.